Hello everybody, I am L.M. Kundu. I am a faculty at the Department of Chemistry, IIT Guwahati, and my research area is bioorganic chemistry, which is an interdisciplinary area of organic chemistry and chemical biology. So, I will be teaching a course which essentially describes the insights into the biomolecules. The title of the course is Essentials of Biomolecules, Nucleic Acids, Peptides and Carbohydrates. Biomolecules are the molecules which are present in living organisms, in biological systems, for example, our body. The examples of biomolecules are DNA, RNA, proteins and carbohydrates. These are the long molecules present in our body. On the other hand, smaller molecules like vitamins, fatty acids, cholesterol, etc. These are the small molecules that are also called biomolecules, they are also present in our body. So, most of these biomolecules or all of these biomolecules are involved in certain chemical transformations in the biological systems. So, in this course we will see how exactly these biomolecules are involved in the chemical processes. Secondly, what kind of chemical transformations occur inside the living organisms due to presence of these biomolecules, and why do they prefer specific biological activities. So, that is one part of the course. Second part is, once you understand the biomolecules, once you know the chemistry of these biomolecules, once you understand the chemical processes that are going on inside the biological systems, then we can alter those chemical reactions. For example, if we understand the chemical reaction that makes a virus active, we can alter that chemical reactions to make the virus inactive. Similarly, we can develop molecules which can cure a disease, which can alter the course of a biological reaction. So, the key topics of this course will be how the chemical structure governs the 3D shape of DNA and RNA, process of DNA replication, PCR, DNA sequencing. Here we can see how beautifully the chemistry and biology make variation in nature. DNA damage, mutations and cancers. What are the factors that induce various kind of changes into the DNA, therefore, that causes the mutations and in turn disease like cancers are formed. Changes in chemical structure of DNA upon damage and how they alter the base pairing properties and induce mutations. Next come artificial base pairings, the molecules that you can develop in your laboratory that will mimic the biological base pairing systems. Proteins and peptides, variation and importance of structures and biological functions. Next, DNA to proteins, how proteins are biosynthesized taking the informations from DNA, how proteins are synthesized using the genetic information. Chemical synthesis of peptides and carbohydrates as well as DNA. Here we can see how we can develop the short peptides, shorter version of DNA as well as the carbohydrates using organic chemistry tools. Then some modern techniques used in chemical biology, the various kinds of chemical tools as well as the instrumental techniques that we use nowadays to study chemical biology for disease diagnosis and biomolecular probes. So, this is the rough course content: the nucleic acids and proteins, synthesis of nucleobases and nucleosides, DNA replication, polymerases, DNA sequencing and PCR, DNA damage, mutations and cancer, DNA to proteins, transcription, translation and genetic code, peptides, sequencing and therapeutics, solution phase and solution phase peptide synthesis, expansion of genetic code, PNA, LNA and molecular probes. Then modern techniques for biomolecules and disease diagnosis, and finally, the structures and chemistry of sugars and carbohydrates and their applications in chemical biology. Thank you.